So what I'm going to talk about today is a algorithm that we developed for detecting m6A methylation sites or peaks in transcriptome, in RNA. So let me talk very quickly about m6A. So Gong in the morning in this session, in this room, actually talked about m6A in bacteria, particularly in DNA. But what I'm going to talk about today is m6A in transcriptome, and particularly in mammalian cells. But still, this m6A essentially is adding a methyl group to adenine in the 6th position of the carbon ring. So why are we so much interested in studying m6A in RNA, particularly transcriptome mRNA? Okay, so this is really due to 2 publications in 2002 that finds surprisingly over 25% of the transcript contain this type of a methylation. And subsequently, while this is the distribution of m6A in a transcript, as you can see, there's a highly enrichment around the stop codon in the 3' end. Okay, so subsequently, due to this study, a lot of people suspect because of this widespread presence of m6A, it carry out a lot of functions. And indeed, a host of research have now shown that m6A are involved in every stage of RNA metabolism, including mRNA stabilization, translation, so on and so forth. And just want to talk a little bit about how this m6A is being methylated. And what's really different between m6A methylation in RNA and DNA methylation is that this m6A methylation is a highly dynamic process. It's actually being methylated through a methyl complex, including 3 proteins. They are METTL3, METTL5, function as a methyltransferase, and a core function WTAP. WTAP itself is a splicing factor. So these are the methyltransferase, and they can be also removed methylation by 2 proteins. Now we know they are FTO and ALK-BH5. So they are demethylases. And in order for methylation to carry out function, it requires the so-called riders. Riders are also a set of methylation recognition proteins. So now we know these YTH domain protein functions, a series of these YTH domain functions are functioned as readers to facilitate subsequent functions like alternative splicing, mRNA stability, and so on and so forth. Okay, but still, there's really a little that's known about m6A functions. So there are still a lot more work to be carried out in this field. And also that we really need more informatics work to understand the function, to predict the function of m6A. So I'll quickly talk about how do we detect m6A. So Gong in the morning talked about using smart sequencing. But for m6A detection in transcription, the widely used technology is MeRIP-seq, or methylated RNA precipitation, immunoprecipitation sequencing. Okay, so MeRIP-seq is very much like a RIP-seq, but it has 2 passes. So in one pass, well, first it starts with the fragmentation of RNA into about 100 base pairs, and then one portion of the samples is subject to anti-m6A immunoprecipitation. And then the other path, and after the immunoprecipitation, the pull-down sequences were subject to sequencing. And the other portion of the original samples will be sent directly to sequencing as the so-called background. So this part is called IP sample, and the background is called input sample. So with these 2 sets of samples, we can use them to detect where the m6A peaks are, and we can carry out subsequent prediction of functions. Okay, so just tell you a little bit about the differences between MeRIP-seq and the chipset. So the main difference here is because the peaks actually now span across axon-axon junctions. So this is not normally what you see in chip, because chips mainly apply to DNAs. And also the peaks now are resides on isoforms. So this is also very unique. In addition, there's a huge variation in the IP sample, input samples, which you don't normally see in chip. So in order to overcome these difficulties or these problems with MeRIP-seq, we previously developed an algorithm called Axon peak that addressed some of these problems that I brought up in the previous slides, and that can predict these sites, and also can carry out the so-called differential methylation, m6A methylation detection under 2 different conditions. Okay, so one of the core, let me quickly, one of the core parts of this Axon peak algorithm is an axon-based paradigm that essentially, puts different axons together to form an axon, and then proceed with the detection of the peaks. Okay, and Axon peak actually uses a so-called C-test. It's a very simple test for peak detection. However, the problem with Axon peak is that it doesn't really consider reads variance. So this is a huge problem in MeRIP-seq. And also, it doesn't consider the dependency of the reads across different sites. Okay, so this is what we set out to address with this new algorithm. So the new algorithm might actually start by first form the so-called axons, and then divide the bins. And after dividing the bins, we can form the models. I can quickly talk about the reads model in one bin. Okay, so we start by using Poisson model to model the reads in both IP and input samples. And then, given the total reads from the input IP sample called T, and the IP sample actually follows a binomial distribution. So we essentially now combine 2 models into one model. So with this, what we do next is so we can build the so-called graphical models to address these 2 problems. One is bin-to-bin dependency. So Z represents the methylation status or peak status. So when Z is equal to 1, it represents a peak, otherwise it's not a peak. And then we build essentially a Markov model there to model the dependency of the peaks. And in addition, so this is that binomial model that we see to model the IP reads, actually given the total reads from IP and the input. And then to model the reads variation, actually one particular thing is this probability P, the binomial P, actually varies across all the bins. So they're not the same, they're varying. But we put a prior on top of all the P's. So essentially we build a Bayesian hierarchical model. We use a beta distribution on these P's across all the bins and all the replicate samples. And with this, actually, we can integrate our P. So essentially, we have a model which is a beta binomial model to model the reads. Okay, so so that's how we really model the bin dependency and the reads variation. Okay, so and then when we have multiple replicates, and we can put all these the distributions together. So this is the joint likelihood function. It's very messy, but so the parameters that needs to be estimated are the set of z's, which are the peak states, and the nuisance model parameters. Okay, so the inference is a little bit complicated in this case, but overall we use a EM paradigm. So just to tell you, we were able to successfully estimate all these parameters and also predict the z. So just to show you the performance. So we start by evaluating the performance using simulated system, basically simulate the reads using these graphical models. First of all, we look at the variance, how the performance. So the performance is evaluated using error under the curve, ROC curve or AUC. And we compare with Axon peak, so with a very varying reads variance. As you can see, the performance of Axon peak, map peak actually is quite robust against the change of the data variances. And then, we look at the number of replicates at different level of variances. So again, these 2 curves are map peak performance. These are AUCs, and they are quite robust. Have a quite robust performance even with small number of replicates. So this is what really what the reality is. So normally we have small number of replicates. And then we look at the data outliers. So some of the replicate, in the replicate, some of the samples could be really, really outliers. So what's the performance of MAP-peak? So as we can see, the MAP-peak also is quite robust against these outliers. And it's also very sensitive to lowly enriched peaks. So this is another scenario that we simulated and tested. Okay. So and then we proceed and we start So how can we really use real data to also measure the performance? So one thing that we did is now we look at these two data sets. One is the so-called knockout FTO. So FTO is a demethylase. So in this case, demethylase FTO is knockout from a mouse cell. Okay, so we have these two sets of data in this data sets, and then in other data sets, these methotransferase, 3 elements of a methyl complex is being knocked out. Each one of it is being knocked out from the HeLa cell. And then the MeRIP-seq samples are measured in all these 5 of 6 cases. So then what we did here is, we take these, for example, FTO MeRIP-seq data, and we swap part of the IP samples with the input sample. So essentially, after this swapping, we should not really see any differences between IP and the input. So then we proceed with MeRIP-seq peak detection. So if we detect any peak, those peaks should be false positives. So using this approach, we can address specificity of the algorithm using the real data sets. So what you're seeing here is the ratio of false positive peaks between MeRIP-seq over Axon peak, not MeRIP-seq, between MACDB over Axon peak. So as you can see here, all the values over these 4 different data sets. As you can see here, all the values are below 1, representing that MACDB actually has a smaller number of false positives. So on average, actually, MACDB has 3 times less of false positives than Axon peak. So these are the results across these 6 different data sets. Okay. So now let me tell you a little bit about the prediction results on these 2 data sets, okay, the real data sets. First of all, in terms of number of peaks, so I'm only showing you the number of peaks in 4 data sets, but overall, on average, MACDB predicted about 13,000 peaks in each one of the data sets, so 13,000 m6A sites, okay, at this cutoff rate, okay. And how do they distribute? So this is the distribution of the peaks in Axon, okay, in these 2 data sets, and in Axon in these 4, in transcript in these 4 data sets. What you can see again is they are highly concentrated around the stop codon in the 3' UTR region, and there are also a big portion of the m6A locates in CDS. Okay, so these are the distribution. We also find actually methylation sites in link RNA. So these are the distribution link RNA. They distribute pretty evenly across link RNA, okay, in these 6 data sets. And these are the motifs. We also proceed with the motif identification of these sites. And these are the motifs, and we see this constant appearance of a GGAC. So this is also being identified as one of the motifs of an m6A methylation in transcripts. So, and then what we did next is we look at the motif, and then we measure the distance between the motif to the center of the peak in each one of the predicted peaks. And then we compare between MAP peak and Axon peak. So what we see here is this is the distribution of the distances. So what we can see is the peaks predicted by mid-peak are closer to the center of the peak. So this is another indication that MAP peak predicted peaks are likely to be true peaks. Okay. So, and then we look at the peak enrichment, essentially the height of the peak, okay? But in this case, it's enrichment between IP and the input samples. And then compare between the MAP peak and Axon peak. So what we see here is this is the distribution of a MAP peak for the enrichment. So the MAP peak actually predicts more peaks that are with higher enrichment, okay? So this is also from another perspective, tells us that the predict peaks are likely to be, true peaks than Axon peaks. Okay. So, and then what we did is we look at the enrichment, and across these 3 cases, 4 different cases. So what we did here, let me back up one step. What we did here is we look at the predicted peaks, the enrichment of predicted peaks in FTO knockout data sets, and then we compare with the predicted peaks in the wild type FTO, the mouse midbrain cells. And then we compare, we calculate the ratio, okay? So in the case of FTO, because FTO itself is a demethylase, once you knock out demethylates, the methylation level or the enrichment should be going up. So this is the average methylation level. This is log scale of, so we compare with 0. So indeed, on average, this enrichment actually is above 1, okay, above 1. So that's consistent with what we expected. So in other 3 cases, what we did is we carry out the same, but for METTL14 knockout, METTL3 knockout, and WTAP knockout. So these are 3 proteins in methyltransferase. So if we knock out these 3 proteins, we should expect methylation level going down. So in these all 3 cases, you see that the average methylation level actually is below 0 or 1. Okay, so that's also very much consistent as what we expected from the experiments. Okay, so we now also have a database. It's called MACDB that stores about 74 different MeRIP-seq data sets, and from 22 experiments. We also have a browser that can help you to really browse these sites. We put a lot of other data sets, including microRNA binding sites, RNA protein binding sites, where you can compare these methylation sites and with these other functional informations to possibly come up with hypotheses. So just to conclude that we have this new algorithm that models this reads variation and reads dependency in MeRIP-seq data, that we show that this algorithm has higher specificity and sensitivity in predicting m6A sites. Okay, so let me conclude by thanking my students and collaborators and the funding supports.